Okay, I think I'll give people just a few minutes, but what I thought I'd start with is going into the models of the medulla and showing you the vasa recta, because it does address the issue that was, let me see which lecture question we're dealing with there. Lecture question 8, which says, to explain how the features of the vasa recta and its blood flow enable the gradient in the medulla to be stable. So I wanted to kind of overlap what we're doing in the lecture with some of the histology and the models that we have in the lab. That way, when you come to the next lab practical, I don't always, as you gather by now, say, what is this structure? What does it do? What is this structure? What does it do? What is this structure? What does it do? But I may ask something like, tell me about the permeability of this structure. Tell me about the osmolarity of the filtrate when it's in this structure. You know, there could be questions that relate to what you're getting in the mechanisms today in lecture. So let me share the screen with you for, this is the medulla in that old plastic model. I'm going to make it a bit bigger here so you don't have so much distraction. So let me see if I can get the annotator going too as well. So what's happening here, and one of the reasons that I've kept this model when we've got the newer ones is because it does take a different strategy to try and illustrate the vasa recta. So I want to remind you that this purple is the thick segment of the ascending loop. For reasons that escape me on this model, this is the PCT, where it's becoming the descending loop of Henle. And then here's that thin segment of the ascending loop. Now, they make it look almost like it goes into the medulla, which it doesn't. The PCT is entirely in the cortex. This is the collecting duct, and as you can see, it's collecting from multiple, and that's what these branches are, from multiple nephrons, not just from one nephron. So the red and blue, appropriately enough, is the vascularization here. And I may even go back and show you the cortex if you think that would help. So the blood that is flowing into what's known as the vasa recta is coming from the efferent arteriole of the juxtamedullary nephrons glomerulus. So as the blood comes out of the glomerulus, if it is from a juxtamedullary nephron, or if it's from a cortical nephron, doesn't matter which, it's at 8 millimeters of mercury. It's very low pressure, and the blood is flowing very slowly because the pressure gradient is small. It's not being driven, it's forced through this. Now, you're going to need that advantage in the vasa recta, because what you want to have happen is the blood to flow very slowly, since the black area here is that interstitial space. And remember, there's a gradient starting somewhere around 300 in the cortex down to 1200 out here, or even a little bit steeper if you're dehydrated. And you don't want to disturb that gradient, because you're using that gradient to pull water out of your thin segments descending loop of Henle, and that water is being picked up into the capillary of the vasa recta here. So you want the blood not to flow rapidly through this. You want it to kind of creep along. That'll allow it to go to equilibrium with the interstitial fluid much more efficiently, and it also won't push the blood. For example, if the blood were flowing downward, if this were part of the capillary, it's flowing downward, it would blur the gradient, as it were, because it would, if it were flowing fast enough, it could pick up some things here and then push them down into here, and you would see a blurring of the gradient. But by going very, very slowly. The other feature, which is really demonstrated well here, is what the Vasa recta have are anastomoses, meaning that there are multiple routes by which, see, there are all these various routes by which you can get into this capillary bed. The anastomoses means that instead of blood just flowing down one side and up the other side, it's also flowing sideways, parallel to the osmotic gradient in the interstitial fluid. So remember, between the descending loops filtrate and the ascending loops filtrate, there has to be about 100 milliosmol difference. You want to have this to be diluted by about 100 milliosmol. If you look at interstitial fluid to the osmolarity of the interstitial fluid to the osmolarity in the descending loop, they're going to go to equilibrium because it's passive processes that are pulling them out. So you're having this guy pump out, this guy losing water, so that the osmolarity climbs as it goes down. The filtrate goes down this descending loop. It climbs as it goes in the interstitial fluid. And those salts are picked up into this vasa recta as well as the water to keep that in a stable fashion. Now you'll notice in this model, which I like because it shows that the vasa recta is not linked to any one, is there a problem? The loop of Henle, although it's only showing one loop of Henle, it also goes across the collecting ducts. So it goes across other loops of Henle and it goes across the collecting ducts, because soon we'll see that under the influence of hormones, water and salts may exit this collecting duct. It's also going to be picked up in the vasa recta, and that water or salt will be returned to the blood through the venous vessels here. So this particular model, this is a patch of vasa recta. It's not they're just making like one patch showing it going across loops of Henle collecting ducts, but it's not unique to one loop of Henle or one collecting duct. They're all jammed in, as you're going to see in the slides, so that it's just going to equilibrium with interstitial fluid that could be from several different collecting ducts and several different loops of Henle. So that's why I like this old model, because it gives you that sense. Now the new model, I'm going to stop sharing this one and start sharing the newer version of this model. Oops, what happened? Let's stop sharing again. Maybe I went too fast for Zoom here. And get this one. It's still coming back to this. Okay, let me close this. Maybe that's the problem. Are you seeing the new model now? Maybe it was just underneath. Or is it?
Yes, I believe so.
Oh, okay. So in this one, once again, I'm not sure why they made this cortical nephron go so deep it shouldn't be. But here you're seeing a juxtamedullary nephron making the descending loop of Henle. And then once again, descending loop of Henle is over here. But once again, I'm questioning how high up the thick segment is. You know, you'll see in your slides, you have quite a bit of thick segment. It's only really way down here that it's a thin segment for the ascending loop. And then it's flowing into the DCT in the collecting duct here. So as you can see, the blood is exiting. Let me get a little pointer instead of this plus, if it will make it. Yeah, the blood is, let's see, where's the best one to pick? This is a cortical nephron. Now, how I know this is a cortical nephron is here's the afferent arterial, here's the efferent arterial, and notice the capillary beds in the cortex. This is a peritubular capillary. The strategy that this model maker took was to just displace the vascular model from the nephron. They don't overlap each other. But in fact, this would be just chock full of PCTs and DCTs and collecting ducts from both types of nephrons. So what would lie underneath this capillary bed, the peritubular, would be PCTs, DCTs, and collecting ducts from any kind of nephron. It could be juxtamedullary or cortical. So let's take a look at, and they didn't do a good job of this here. They didn't really do a good job of showing the efferent. Let's pretend there's a glomerulus here because they're not showing this as the efferent. But it's coming out of an efferent and it's fed, now it would be, let's say it's this one. We'll just pretend that's connected up to here. This is a juxtamedullary nephron, and its efferent then flows into this long capillary bed, which is the vasa recta. Once again, they've displaced it. They're not showing what's lying underneath it, which would be the descending and ascending loops of Henle and collecting ducts. But it could be many different ascending and descending loops of Henle and many different collecting ducts. So there's no one-to-one linkage between a particular vasa recta and a particular loop of Henle. As the blood comes downward into the medulla, it goes isoosmotic with the interstitial fluid. So up here, it would be 300. Down here, the osmolarity of the blood would be 1200 or even higher. To help it maintain it, it flows very slowly through this vasa recta, and it has anastomoses. So these anastomoses are showing, multiple ways to get over to the draining venous side. And this vasa recta, as it's traveling, is picking up the water that is exiting the descending loop over here into that space and picking up salts that is being pumped out of the ascending thick segment. At the same rate the salts are being pumped out and the water is leaving, it's being picked up by the cleanup crew here, which is the vasa recta. So hopefully this is making sense with the caveats I gave you, like they don't show a good efferent arterial. They look like it's coming right from the cortical radiant in this model. Maybe this one's better. Here's an efferent arterial. And it's making a vasa recta, but then whatever reason the model maker decided to not do the anastomoses and connect it to the venous part. I don't know why they chose to do that, but you can see this feeding into the vasa recta. All right, any questions? Let me see what's in chat. There might be something in chat. Post a key to the model since this one has numbers. Let me get rid of this thing. Let's see if I can read numbers very well on this. I think we probably could, but it is, like, for example, that's a D. So sometimes I'm not going to be sure what they are trying to point out with a D because I don't actually have the key to this model. I could ask Kevin, like I can't tell if that's a C or what that is, but I could ask Kevin to send me the key. This is a brand new model, so he should have it. Generally speaking, though, here's the glomerulus, right, with the glomerular capillaries. Here's an afferent arterial feeding them. And then here's the short, way too short for reality, PCT, and for some reason they have it going into the cortex a bit, which it doesn't really do, or not very far, and then becoming the descending loop, the thin segment of the ascending loop, and then the thick segment of the ascending loop, and then it becomes the DCT. And you can even see how it swings by the afferent arterial and becomes the macula densa to detect, which detects the salt level. It should be at 100 milliosmol in here. And if it's not, then intrinsic factors would slow filtration, slow the movement of filtrate through this, giving the PCT time to pick up more salts and hopefully bringing this back to 100. Now, ultimately, it leaves here at 100. Unless there's hormones that are released, you're dehydrated, you need to conserve water, or you have salt imbalances, you need to adjust salts, it will just leave this at 100 milliosmol. So that's a single collecting duct. The other point that's nice here is you can see this, if this is a cortical nephron, this is a juxtamedullary, then the filtrate made by each of them, in theory, this filtrate ought to be about 300 milliosmol since it doesn't have the loops of Henle, despite whatever that is meant to be. You can even see there's very little thin segment. This should be largely in the cortex, not all the way down here, but that would mean it's going to leave somewhere close to 300 milliosmol. It's not going to be 100 milliosmol because it doesn't have the loops of Henle. So you're going to mix and match together in here the filtrate from both the cortical and the juxtamedullary. This one, say, leaving at 100, this one 300. They're all mixed together in the collecting duct. And then if hormones are present, you can extract water or salts from both of these nephrons by having the hormones work on the collecting duct. I'm not sure, other than there's no letters on this to point out anastomoses, but, we can do the best we can. I think that I can't really read that letter either. So I can try and, see what I can do with the letters and the keys for the key on this. Okay, so at this point, unless there's other questions, I think screen sharing stopped. See if a chat, there's anything else. Okay. Let's go to looking at some of the slides. So I did kind of rapidly walk you through these slides last week so that you could get started on this, but I know a lot of you couldn't or weren't able to be at lab the day I did some of this. So I am going to go first to point out, let's not do the bladder yet. Let me screen share you have this, but I'll screen share the study guide first. Hopefully this share will work right away. There we go. So let's start just looking at the kidney cortex and the medulla. And I did walk you through a lot of the cortex. I have the macula. Yes, I have the macula densa. We went through that using labeled slides that were from the study guide I put together. These aren't necessarily the same exact pictures you have, so I would recommend you draw the pictures you have and add labels to them, and you can use this as a reference. There's also good pictures in the histology book, in this case, I think better pictures, photographs, showing you the macula densa. And they are actual photographs, not drawings, but they have drawings as well. If you want to go in de fiores, that should help you with this. Now, I did tell you didn't have to tell PCT from the DCT here, partly because there is a color temperature difference, staining difference. This is a brighter red, but I think in the photographs they're a little bit hard to tell. So let's look at the medulla, and now that you know something about it, the vasa recta has blood in it. That's how you'll know you're dealing with the vasa recta. You'll see these ghostly blood. And you can see there's no one-to-one. In fact, you have vasa recta, and it's probably another vasa recta, and another vasa recta here. And here's another one. And then squeezed in between here, we have some very, very flat. I wouldn't use this one because it's all, it's been cut at an odd angle. But you see they're really flat squamous cells here, and they have this kind of greenish color. And as you can't see that lumen very well, but over here you can. So here you're seeing a very flat cell, an empty lumen, and another very kind of greenish, very, very flat cell. So this is simple squamous. Simple squamous is going to tell you that you're either the vasa recta, like the simple squamous here, or the descending loop of Henle. And you'll say, well, what about the thin segment of the ascending loop? I won't worry about that. We're doing all these drawings fairly close to the cortex. All the pictures I took, they're not way down deep at the tip of the medulla. So we're not going to be seeing any of this thin segment of the ascending loop. So if you're seeing squamous, it's going to be black and white. That's descending loop of Henle. My dog is going a little bit crazy over here. Hey, Kyle, can you take him out? Maybe he has to go. So there you have the descending loop of Henle. So how do you know the ascending loop? Well, remember the thick segment are simple cuboidal cells. And since again, we're fairly close to the cortex, those simple cuboidal cells are going to have really round nuclei like this. And there are two things that could have simple cuboidal. One is the ascending loop and the other collecting ducts. In this case, in this picture here, I don't have a good collecting duct, and it's possible one of these over here is, but they're kind of out of focus. So this brighter pink is going to tell you that this is a ascending loop. If it were a collecting duct, the color looks quite dull. For example, see the dullness of this color? That's probably a collecting duct, but because it's out of focus, the other big sign it's a collecting duct is the inner edge doesn't look blurry. It looks fairly sharply focused, but it looks like I focused for this part of the slide, and this over here is a bit out of focus. So unfortunately, I don't have a good focused slide of a collecting duct, but these are collecting ducts with a very clean inner edge. They don't look blurry, other than whole pictures out of focus in this. So let's take a look at your photos. I'm going to stop sharing this. And remember, the ones I'll put on the test will be from your drawings, your photos, by and large. Okay, let me go in there. Urinary slides. And let's go to a high power of the medulla. So, okay, I'll leave that one open. I'll close this. And let's share this one. So this is from the slides posted that you're going to draw things from. So you see the nice sharp edge here? These are very cuboidal cells. That's a collecting duct. See the blurriness, and it is kind of a brighter red as opposed to a duller red. The blurry kind of this, it's going in and out because of the, there's a curvature, and when they cut, some places cut through cells, other places cut through the lumen. So don't be confused by that. This is a thick segment of an ascending loop. This guy, see the flat nucleus, the dark line of this edge here, and you can only really see just kind of the dark line here. It's hard to see nuclei, but you can see there's kind of trash in this. It doesn't look like the ghostly red blood cells. So this is a descending loop of Henle. Here's probably another one, although I'm struggling to see the other side here. It could have just been collapsed down. That's probably what's happened. So you have one side of the descending loop and the other side of the descending loop right there. Here, on the other hand, you can see the flattened cell that's kind of a darker line, and that's probably another descending loop. So what is missing here seems to be the vasa recta. My guess is we don't have a very good view of a vasa recta, but this might be one. That might be red blood cells. It's not a very good view of a vasa recta in here. So I'm hoping you get the idea looking at this, that you don't have this organized, here is a descending and next to it is an ascending. Next to that's a vasa recta going down, vasa recta going up, that the drawings imply. You have everybody just crammed together. And the reason you can get away with that is there's very little, but there is interstitial fluid. And what you're trying to influence is the osmolarity of the interstitial fluid with the ascending loop, and then having that osmolarity affect the descending loop. Having salts pumped out into it, yes, and water exiting into it, but very quickly the vasa recta is picking those up at the equal rate at which they are being ejected. Okay, let me see if there's any other slide of the medulla worth looking at. Here's the other slide of the medulla. And just, as long as you can recognize it, you're going to draw one or the other, or both, whatever you wish. And I stuck two in here because this one shows you quite a bit of vasa recta. See all the blood? So all these little things that look like tiles or red blood cells, going through the vasa recta. It does show good, although cut and cross section rather than longitudinal cross sections through the ascending loop, the brighter red. And occasionally you find areas like this without blood in them, but these flat cells on each side, those are the descending loop. This is probably transitioning to becoming flat enough to be a descending loop. But what's missing here is I don't really see any collecting ducts in this view. That's for sure a collecting duct. Possibly it's probably ascending. It's too bright red. So you need to get that search image for the color, what's in the lumen, and the shapes of the cells. So are there any questions or anything you're struggling with from the cortex? You can just put it in chat. I think I went through the cortical slides last time. Okay, so I'm going to leave the kidney and we're going to jump forward because I believe what I put down is today, I move things up. Today you don't have to turn anything in, but by Thursday, you will need to turn in the bladder and ureter as well. And so since we're going to talk about that in lecture, I don't want to spend time in lecture talking about their histology. I think I did to a certain extent last week, but we hadn't gotten there in the lecture. So let's talk about the histology again. I'll show you those slides again and try and relate it to what's going on in these, which I think I did. So I'm probably repeating myself if you feel like you don't need it. So Thursday at 5, you're turning in the drawings of the kidney that are outlined in the study guide and the drawings of the bladder and the ureter. There are more photographs posted than there are drawings. So it's up to you and your judgment to have a nice picture showing all the required labeled structures. So one of the things that is required to be labeled are the mucosal membrane layers again, and their subtypes of tissues should be known. So let's start with the bladder, and I'm going to the higher power picture of the bladder. Ureter, not bladder, I misspoke. This is a ureter. It's from an animal. Sorry, sometimes my camera gets in the way of itself. Anyway, here is the lining. This is where the urine would be flowing from the kidney towards the bladder in this lumen. And lining this lumen are transitional epithelia. So this particular mucosal membrane, they don't have specialized cells or glands that release mucus. So these transitional epithelia have to release a small amount of mucus. it's not the amount of mucus you would see in either the digestive tract or in the respiratory tract, because you utilize flushing as a main way of physically trapping and pushing out pathogens that might be trying to go upstream from your urethra to your, hopefully that's not being shared, upstream from your urethra towards your kidney to prevent you from getting a kidney infection. So this is one reason why we're trying to work with my dog about this too. You don't want to retain urine for long periods of time. So you may, it's much healthier to urinate on a frequent basis than it is to hold urine in your bladder, even if the bladder can do it, because you utilize flushing more than mucus. There is a little bit of mucus released by these transitional epithelial cells, but there are no glands to release mucus, and there are no specialized cells like we'll really see in the digestive tract that make lots of mucus. So that is the epithelial layer. Now underneath it, and I don't expect you to distinguish the lamina propria from the, let me see if I can annotate this somewhere, from the submucosa, because there's no real glands. There's probably lymphatic follicles in there, but none of our slides have I found one. So right here, right under that would be a lamina propria, but I don't think you could really, with confidence, tell it from the submucosa. So I picked this area because it has this fold, and you can see the submucosa, and I'm going to draw a line. Now, the line's a little high. Maybe I'll erase that. The line's a little straight, and maybe is a better way of putting it. Where the muscle layer, I'm trying to follow the muscle layer here. So this sort of region here that's going along the edge, that's submucosa with lamina propria. Now it's possible this redder color is lamina propria, because see how it follows it up into the fold here. And it would be potentially redder because it's very vascularized, for those guys who missed it on the practical. Highly vascularized with immune cells. That is not follicles, but like mast cells, things like that. That's what's rich in areolar or connective tissue. Whereas this is more fibrous connective tissue, and the pink you're seeing is probably a lot more collagen that is structural to hold this layer to the muscle. So then we have the two layers of muscles here, the circular and longitudinal. And just like in your digestive tract that we're going to start, this is visceral smooth muscle. So in the respiratory, it was multi-unit smooth muscle, but this is visceral smooth muscle. You want it to be visceral because what you're going to do, especially in the bladder, is you're going to stretch it. If you stretch multi-unit, it's going to contract. You don't want that to happen in your bladder. You want it to be visceral, so when you stretch it, it automatically relaxes. Now, in the ureter, you use a lot of gravity to move the urine from your kidney to your bladder, but these muscles help as well. So you have a circular and a longitudinal. So one layer goes around. If I looked at a cross section, one layer would go around, that's the circular, and the other one would go up and down the length. That's the longitudinal. We'll talk more about this in the digestive tract, but one of them does peristalsis and the other one does segmentation. Basically, the circular kind of clumps down on it, squeezes in a circular way, as you might imagine, kind of like what happens in vasoconstriction, and they call that segmentation. And then there's a wave. Now, they can do this because this being visceral smooth muscle, it has pacemakers and gap junctions that allow the muscle to be synchronous in its activity, kind of like the heart is. So there are specialized muscle fibers that we won't name that are acting as pacemakers, and there's gap junctions between all this visceral smooth muscle that allow them to act as a sheet of cells and They do respond to hormones, but mostly they are reflexively contracting. So finally, you get to the outside here, and this is going to be adventitia, not serosa, because this is retroperitoneal. You see adipose cells here, and they're cushioning the the structure, because it's really just connected up and embedded in musculature. There's no bony protection for the ureter. There's a bunch of blood vessels here. Now, I did get people answering that the areolar is used for cushioning, and I know that was a function of areolar in 103A. I don't think it's a main function here. It's very loose connective tissue, so there is a lot of interstitial fluid in it. I suppose the water can cushion it. But in this case, unlike what you'll see in the digestive tract, there's not adipose embedded in the areolar here. So I wouldn't necessarily pick that I accepted that as an answer, but as a very good answer to why you want areolar here. You want it here because of its high level of vascularization and rich in mast cells to help fight infection. All right, so I spent quite a bit of time on this slide. I'm going to stop sharing it. Hopefully that means all of that will disappear, which it probably will. Okay, now what about the bladder? Let's see my higher power picture of the bladder. Do I only have 2 at 40? Maybe I only have 2 at 40. I didn't take a higher power picture. Yeah, I don't know why I don't have a 100x at least. Doesn't matter which one. Oh wait, 400, I do have one here. Okay, just being blind. Maybe I'll open both. That's okay. Just open this one. I think you can see enough in this one. So now when we get to the bladder, let me make this a bit bigger. So let me get my annotation. So once again, there is transitional, did I get this? Transitional epithelia that is lining this. So it's the same lining. And again, so these guys can be really, really stretchy. Clearly the bladder has to stretch in order to do its job of storing urine. Now, it also has to reflexively, and we'll talk about the reflex in lecture next time, contract to expel the urine when you urinate. So we don't have these in the slides, but there is a sphincter, a set of sphincters at the opening of the urethra, and we'll talk more about those in the lecture. So transitional, really good because it's nice and stretchy. It's also stratified because, once again, this is an area that is subjected to bacteria, especially even more than the ureter, because the urethra has normal resident bacteria. So you want all this stratification. Now, yeah, it makes a little mucus, but not a whole lot of mucus. There aren't really glands, and there are not specialized cells. So underneath that, there is a lamina propria. Again, I probably would not ask you to point it out. You might be able to kind of see an area here, but it's hard to say. So instead, I'm going to make the same sort of line, hopefully, if I can control this well enough. So this is the submucosa. And once again, you don't have glands in it, which I also got on the practical. I got submucosa, subtype of tissue, and I was looking for fibrous connective tissue, and I got glands, which exist in it, but not always. And the main tissue subtype is going to be a fibrous connective tissue because of its job of holding the muscle to the epithelial layers, providing blood also, but the lamina propria is even more important for that. And it can have lymphatic follicles in it because it's a mucosal membrane. Yeah, sometimes there are glands, but for example, in the bladder, there are not. in the ureter? There are not. Glands are not required to be a submucosa. Okay, so once we get through that, again, fibrous connective tissue, you get to 3 layers of muscles in the muscularis externi, which you only had 2 before. So the 3rd layer is obliquely, oops, I can't make this a different color format. Maybe I can. Oh, let's see what happens. Oh, yeah, oblique orientation of the 3rd layer, and that layer is called the detrusor muscle because it helps the bladder collapse down. So know that there are 3 layers, but I don't expect you to identify them. Now, with this higher power picture, I don't have the outer edge of the bladder. Now, the bladder kind of projects up into the abdominal cavity. So when you look at the models, you'll see that. The part that's poking into the cavity has serosa. The part that's in the pelvic floor has adventitia. I think the only thing our slides show is the serosa area, so you don't really get to see much there. There's not much there. So you just have to know that's serosa. Looks like there's something in chat that's new. It is transitional, yes. Okay. Let me get rid of chat, which I can't do without moving this. Okay. So when we get to this in lecture, it doesn't want me to do this because I have to go back to here. We are going to be talking about the 2 sphincters at the opening of the urethra where it bleeds into the bladder. So you won't see them in the slides, but I would know that you have an internal sphincter. That internal sphincter, I'm going to again write something here, at the urethra, so I'll just say at urethra, the, I don't think you actually know which layers, but I would assume it's the circular layer. These form the internal sphincter. So I'll just put INT, internal S. So when you have, when you go to urinate, the internal sphincter is involuntary muscle. It's visceral smooth muscle, which, by the way, I should have said again, just like in the ureter, the bladder, this is all visceral smooth muscle. And it's innervated by both the sympathetic and parasympathetic nervous system. When we go to urinate on Thursday, what an invitation, what we'll see is that the things are kind of flip-flopped from what you're used to. When you're at rest, the sympathetic is activated and causes that sphincter. This is a sphincter. So the sympathetic is excitatory on the sphincter, and at rest, the sympathetic excites the sphincter, but it relaxes the rest of this muscle. Sympathetic is inhibitory on the rest of this muscle. Oops, I'll just say inhibit. What that means is that you are at rest able to keep relaxing your bladder. Now, the relaxation as it fills is due to that stretch response. As more comes in, you don't need to have more sympathetic. It's just the visceral smooth muscle allows it to relax because that's its nature when it gets stretched. And you're using your sympathetic to prevent excitation of this muscle and opening of the sphincter. You want to excite that sphincter, relax the detrusor muscle so that you don't urinate. This is reinforced once you are beyond infancy by an external sphincter, which is voluntarily controlled. You've probably known that if you've been unable to pee, because you're in a talk or a lecture and you really, really, really have to, you can just feel yourself clamping down with your pelvic floor muscles, and particularly with your external sphincter, which is skeletal muscle. Before you're, I don't know what age of infancy, but when you're an infant, probably before you're 1 or 2, you don't really have good control of that external sphincter, and your internal sphincter and your parasympathetic reflex will drive your urinating. So when you go to urinate, you suppress your sympathetic, you activate your parasympathetic, and that allows you to urinate. So that's a little foreshadowing of where we're going to cover urination reflex, and we'll be finishing up the formal part of the chapter on the urinary system, but it's going to overlap very much with the water balance and electrolyte balance. So when we come back on Thursday, we're not really ever leaving the urinary system, but we're going back to some of the hormones like the ADH and areas of the brain that control water balance. So let me stop sharing that. And see, I don't think there's anything else, but if you guys have some request, there are low powers of the bladder in there. And you'll say, do I have to draw the low and the high? Not necessarily, but if I show a low power, maybe I will just share the low power. Even this doesn't have the serosa on it. At least you should be able to recognize at lower power all those things I just showed you at higher power. So if I put this one on here. So you can see in this bladder, lots of muscle. It's quite a muscular organ. This is all smooth muscle. Now, I won't ask you to tell me which of these is circular, which is protruser, and which is longitudinal. I wouldn't ask you to tell that, but I would know that they're all there. There might be a little tiny bit of adventitia over here. I don't know. And then you can see the transitional. I'd be hard pressed with this magnification to ask you about the submucosa or the lamina propria. It's probably right here. I mean, this is all submucosa probably. But I'd probably be happier to ask something like that on the higher power picture. Okay, questions? Anything in chat? Anyone want to be in a breakout room? You're welcome to do that as well. So remember, all of these slides of the kidney and bladder and ureter are due on Thursday at 5, the slide drawings. On Thursday, we are going to be doing a clinical test called a urinalysis, and you're going to be doing it by watching Kevin do it. I suppose maybe in the future we could, maybe next semester I'll try and see if we can send things home so you can actually test your own urine. If we got real excited about it, we could even do that. Some of the things like the test strips are sensitive to the air, so they can't be packed up, pre-packed up and that kind of thing, and left in the air because they go bad. So other than that, you can watch him do it, and there's a handout you're going to be filling out. The information for the table and all the conditions and what's normal and what's not normal is all in the lab manual. So you are basically transcribing stuff from the lab manual and learning that stuff for the urinalysis part of this. I think I put a couple other videos that go beyond what Kevin does, in the sense that he doesn't really explain. These explain more. And they also look at a few of the things that you would have been doing in the lab, but Kevin doesn't do for you, which is to make a slide of your urine and look at casts and the salts, crystals that can be present in urine. And he doesn't do that. Maybe I shouldn't say obviously, but he doesn't. But the videos do it. And I do expect you to watch those and know what's in those videos as well. So that's what will happen on Thursday. And we will need to finish that Thursday. It won't be due to the following Tuesday, because then we're going to be doing a couple Physio X. These are software that should be available through your mastering. So you'll have to access that through mastering. We will work on them together so that you get the answers to those handouts. Okay, if you don't have any questions, I will let you go. And you've got about 45 minutes to work on your slides or work in groups if you want. And just shout out and get my attention, because I'll probably go and have some lunch and then get on another screen, but I'm here. Okay, I also will post the key to the lab practical. I haven't done that yet, but I will post the key. And I want to give you a heads up that I'm working on right now, getting Kevin to take some pictures of things I missed for the models of the digestive tract when I went in to take pictures, because they were in the tutor center and I couldn't get access to those. So he's getting those for me. And so I will be putting an updated study guide up with digestive stuff. If I'm sure you're all looking forward to that already, but I'm trying to work on that for you. Okay, so just give me a shout, and if you breakout room, let me know that too. All right, see you on Thursday otherwise.